So this is the last chapter, chapter 15, Genes and Proteins. You'll see that the textbook is a lot more detailed than what I do in the PowerPoint. You only have to know the information at the level of the PowerPoint. So proteins are what are built from the DNA, and proteins are the workers of the cell. Protein synthesis consumes more of a cell's energy than any other metabolic process. Proteins account for more mass of a cell than any other component, with the exception of water. And nearly every function of a cell is performed by proteins, and proteins are used to build the other molecules, the lipids and the nucleic acids and so on. So proteins are really, really central to cells. So here's DNA. DNA codes for protein. A gene would be a section of the DNA that tells you how to build a protein. Here are two particular proteins. And so I looked them up. There are 20,000 different proteins in our genome. And the first one is interleukin-2, and it's part of our immune system. It signals to our white blood cells to produce certain kinds of cells to fight an infection. The other one is a mouse urinary protein. So a male mouse would secrete a pheromone, and this stabilizes the pheromone, so a female mouse would know that the male mouse is in the area. Okay, DNA to RNA is blank, RNA to protein is what? Okay, so this is part of the central dogma, the central idea of biology. DNA to RNA is transcription, and RNA to protein is translation. Answer B. DNA is ATG. The complementary mRNA transcribed is, okay, so A matches U, so UAC. And what's this molecule here? It's a molecule with an amino acid on one end and an anticodon on one end. Okay, it's called a tRNA. And what's its function? Okay, so it inserts the correct amino acid into the polypeptide chain based on the mRNA codon. So it reads the mRNA codon with its anticodon. It looks like an L. And on the other end, it inserts the corresponding amino acid. Here's some good videos that will help you. Besides Amoeba Sisters, the University of Utah has a really excellent website. Okay, so there are 20 different amino acids, and just kind of so we remember that an amino acid is made of an amino group, our carboxyl group, a central carbon, a hydrogen, and then 20 different side chains. And by putting those amino acids together in different orders, as specified by the DNA, you build all the different proteins. Okay, so the central idea of biology is that DNA goes to RNA, that's transcription, and RNA goes to proteins, that's translation. All right, this is just showing that when DNA separates, it can be used to build RNA. And then that RNA is processed in eukaryotes. Something called introns are cut out. We'll talk about that in a bit. And after it's processed, that RNA leaves the nucleus and goes to the cytoplasm. And in the cytoplasm, translation takes place on the ribosome. And these tRNA molecules read off the mRNA codon and insert the corresponding amino acids. Okay, I have a lot of exercises on this. After you do the exercises, because this is the last lecture, I have the answers to the exercises. So try to do them, compare RNA and DNA, and then look at the next slide. Okay, so here are the bases of RNA. Here are the bases of DNA. Is it single or double-stranded? What's the sugar group? Is it ribose or deoxyribose? Okay. If you have an A on DNA, it's going to bind to a U for RNA. If you have an A on DNA, it's going to bind to a T for the corresponding DNA. Okay. And T on DNA always binds to an A. Okay, so there are 4 different nucleotides, and a 3-nucleotide sequence is called a codon. So it's like a 4-letter alphabet. And instead of saying there are 3-letter words, we say there are 3-letter codons. Since there are 4 different nucleotides and a 3-letter word makes a codon, there are 64 different codons. We'll show you the genetic code in a minute. Okay, so here's the genetic code. It doesn't matter whether you're a mouse or a mushroom or a plant or bacteria. UUU always specifies phenylalanine. They always show the genetic code, and I'll show it in terms of the messenger RNA. But you could say, well, AAA in the DNA coded for UUU, so you could write it in the DNA language, but by convention, I'll show it to you in the mRNA language. Okay? The genetic code is redundant, so if you have CU, doesn't matter what the last letter is, you always have leucine, because there are 20 amino acids, but there are 64 codons. Okay? So proline is coded for by CC, and it doesn't matter what the last letter is. Okay? Okay. You don't have to know what they stand for in terms of MET is methionine, but you should be able to recognize the sequence of these as being amino acids. The first protein, or the first amino acid in a protein is always methionine. Translation always starts with AUG. That methionine sometimes can be cut out after the protein gets made, but in translation, you always start translating with methionine. Okay, when you're ending translation, translation always ends with a stop codon, and there's a protein release factor that goes in instead of an amino acid, and that protein release factor says to the ribosome, hey, stop translating and release the finished protein. So this is just showing that if you have a couple deletions and everything moves over, it's going to mess up all your amino acids after that deletion. Okay, AGC is serine. AGC represents serine. This is the messenger RNA. But now you deleted 2 of the bases from the second nucleotide, from the second codon. And so now everything's shifted over, so after the serine, all the other amino acids are off. So a frameshift mutation is going to be really a serious mutation. It's going to create a non-functional protein. Okay, we probably already did this in lab, but unlike animals, where if you have an extra chromosome, it causes things like XYY or triple X or Down syndrome, plants sometimes get extra chromosomes with no harmful effects. In fact, plant breeders sometimes purposely build plants with extra chromosomes because it tends to make the flowers and fruits bigger. So they say kiwis are hexaploid, they have 6 of every chromosome, but strawberries do them one better, they're octaploid, they have 8 of every kind of chromosome within them. Bread wheat is hexaploid, it has 6 of every chromosome, and so a lot of common plants that we use have more than the diploid number of chromosomes like humans have. Okay, transcription. Transcription is making DNA into mRNA. And so there's an initiation where a promoter region of the DNA has the RNA polymerase bind to it. There's an elongation phase. That's what we think of when we think of transcription, where A, U, C, and G match up with the DNA. And finally, a termination where the RNA polymerase releases the pre-mRNA. Okay, so this is showing the elongation process. This is what I'll have you model, where there's a C in the DNA, so the DNA is blue on this side, the corresponding DNA on this side. A helicase enzyme has already broken up those hydrogen bonds. So now a G goes for mRNA to match a C, a U matches an A G matches a C, a U matches an A, and if there's a T, it's matched by an A. Okay, so you're building the mRNA one letter at a time based on the sequence in the DNA. Okay, we can go through it. If DNA is GCC, it's going to be CGG. AGT, it's going to be UCA. I'll let you do the rest. So CGG, UCA, we'll look at that. CGG, this is the genetic code. CGG, arginine. Oh, I don't remember what the next one was. If you don't write it down, you forget it. But anyways, follow it through. CGG was arginine, UCA is serine, and you'll have to do one of these on the exam, I'm sure. Okay, so initiation, the big outer tan molecule is RNA polymerase, and RNA polymerase binds to DNA in what's called the promoter region. So the promoter region is before elongation starts, before you start matching DNA and RNA nucleotides, and there are certain sequences of DNA, and it says it's negative 35, so it's that many bases before elongation starts, and certain sequences tell the RNA polymerase, hey, this is where you want to bind. And eventually, on the other side, after elongation, there are certain sequences of DNA that say to the RNA polymerase, hey, this is where you get off. We've finished making the RNA, and now unbind from the DNA and release the RNA. Okay, and this will go into more detail than I went into. So if you want to watch it and get the idea, here's the DNA and RNA polymerase is building the RNA, but don't be concerned with all the details. Okay, in a prokaryote, transcription and translation are happening simultaneously. So the green represents the DNA, the RNA polymerase is churning along, and the RNA polymerase is making messenger RNA. Okay? There might be more than one RNA polymerase going along all at the same time and making strands of messenger RNA. And then similarly, more than one ribosome bind to the RNA at a time to make the protein. So if something might be harmful to the bacteria and they had to build something to detoxify alcohol or break down something in the environment, it's a way to ramp up protein production pretty darn fast by having the RNA polymerase making mRNA very quickly and having all these ribosomes make protein very quickly. Okay, you don't get quite as fast production of a protein in a eukaryote because you have a separate compartment for transcription and translation. So transcription is going to happen inside the nucleus. The finished mRNA will move out of the nucleus into the cytoplasm, and in the cytoplasm, the ribosome will bind, and that's where you'll have translation. You can still get polyribosomes, so you'll get more than one ribosome at a time doing translation. All right, one thing you have in eukaryotes that's not found in prokaryotes is RNA processing. So one of the big surprises in the 1970s that led to win a Nobel Prize is the discovery of introns, that within our DNA are non-coding regions. Exons are the expressed regions that become proteins, and the introns are right in between that. So we'll show you that in a minute, but let's just say you have a chocolate chip cookie recipe, and maybe it says, well, you could substitute caramel chips for the chocolate chips, or you could add pecans, or you could add walnuts. So that same recipe could be about 4 different kinds of cookies, depending on what you add to it at the end. That's called alternative splicing, and one of the surprises of the Human Genome Project is that we only had 20,000 genes. People thought we'd have closer to 100,000, and this alternative splicing of pre-mRNAs into mRNA is partially what accounts for that. So here's the primary RNA transcript, or the pre-mRNA, and so this was built directly from the DNA. These are the exons, the parts that are going to be made into protein. These are the introns, the sequences that are going to be cut out. And so here they splice together exon 1, 2, and 3. But you could see if there were directions to say, oh, skip exon 2, then you don't add the walnuts. So you get a different protein from the mRNA if you don't have exon 2. And this is just showing that even in single-celled eukaryotes, there is a splicing step. So mRNA splicing came about very early in development of eukaryotes. All right, let's move on from transcription to translation. So in transcription, you're going from RNA, from DNA, deoxyribonucleic acid, to RNA, ribonucleic acid. But you're still in the language of nucleic acids. You're still in the language of nucleotides. Now you're translating. You're going from the language of nucleic acids, from nucleotides, into amino acids, into proteins. So it's called translation. Okay? Okay, and a key molecule in translation, it looks like an L, and it's called transfer RNA. So just like there are RNA polymerases that are building messenger RNA, there are RNA polymerases in the DNA that are building these transfer RNAs. And these transfer RNAs are this L-shaped RNA molecule, and on one side is the anticodon that matches the codon on the mRNA, and the other side it inserts the corresponding amino acid. So the codon of the mRNA would be UUC, and UUC is going to code for phenylalanine. Okay, and this is just showing that in the end, you're going to have a chain of amino acids linked together. A typical protein like hemoglobin would be 146 amino acids long, all coded for by the DNA, DNA into mRNA, and the mRNA into protein. Dehydration synthesis, water comes out, and the carbon from the carboxyl group joins the nitrogen from the amino group. With translation, like transcription, we call it initiation, elongation, and termination. Okay, so this is just showing those processes. So in initiation, the small subunit of the ribosome joins to the mRNA. The mRNA has left the nucleus and is now in the cytoplasm. AUG, coding for methionine, always starts the mRNA. So it always starts a protein, but sometimes it gets processed and cut out later. So here's a tRNA. The UAC is the anticodon for the AUG, and it carries a corresponding amino acid, methionine. I once had a faculty member who said, tRNAs are like trucks. They only carry one load, and that load is specified for by the anticodon on the front. Okay? Then in the second step, the large subunit of the ribosome comes in. The next tRNA comes in. This time it's carrying phenylalanine, which must be specified for by UUC, and the anticodon AAG binds to the UUC. Okay, the ribosome slides along. The methionine and phenylalanine hook together. Once they hook together, the previous tRNA comes out, and then the next tRNA comes in. And the next tRNA carries its amino acid, and then the phenylalanine is going to join to the next tRNA, and this tRNA is going to go back to the cytoplasm to get reloaded with another phenylalanine. Okay, eventually translation will stop, that polypeptide will dissociate, and it will fold up into a functional protein. If you stop protein synthesis, you kill an organism. So tetracycline, for whatever reason, it interferes with tRNA on bacterial ribosomes, but not eukaryotic ribosomes. So if you've had acne, you might have been prescribed tetracycline, and so it kills bacteria, but not eukaryotes. Apparently, eukaryotic ribosomes are slightly larger, and for whatever reason, it doesn't interfere with our protein synthesis. All right, let's compare transcription and translation. Define them. What do they start at? What do they stop at? What's a key molecule? Okay. All right, so DNA to RNA is transcription. RNA to protein is translation. Transcription starts at a promoter region. Certain sequences in the DNA tell the RNA polymerase where to bind. Translation always starts with an AUG codon. Okay, where does transcription stop? stops at a certain sequence of DNA where the RNA polymerase detaches, and that sequence is called a termination sequence. In translation, there are 3 codons that are called stop codons, and they tell the cell to put in a release factor protein, and the release factor protein makes the 2 subunits of the ribosome break apart, and the polypeptide gets released. The key molecule in transcription is RNA polymerase. That's what makes DNA into RNA. The key molecule in translation is tRNA. That's the transfer RNA that reads the mRNA and puts in the corresponding amino acid. Sometimes people tell me the key molecule is the ribosome, and that's also a good answer, although I think a ribosome was made up of more than one molecule. All right, protein synthesis in prokaryotes versus eukaryotes. Okay, how do they compare? Okay. Is there a nucleus? Are there introns? Is it inhibited by tetracycline? Okay, so by now you should know that prokaryotes don't have a nucleus, eukaryotes do. We learned today that introns are found not in prokaryotes, but they are in eukaryotes, and that tetracycline stops protein synthesis in bacteria, but not in humans. All right, so here's a really good site, again, the University of Utah website, and it will have you practice DNA to mRNA and then mRNA to amino acid. Okay, because all cells share common ancestry and the same DNA language, you can do genetic engineering and move DNA around between unrelated organisms. And I think they used a gene from a jellyfish, at least in the yellow fish, to make it glow. And so it reads the jellyfish gene and makes that fluorescent protein and makes the fish glowing. Okay, you can click on this and it'll tell you more about what genes they used. Here's a more significant use of genetic engineering. So before genetic engineering, they would take pigs that were slaughtered and they would buy up the pancreases and extract the insulin from pigs. Now instead they have the sequence of a human gene and they cut out that human gene and they put it into an E. coli bacteria because we are all related, even humans and E. coli. That E. coli bacteria reads the human DNA and it makes human insulin. They call it humulin to show you it's human insulin that was made in E. coli bacteria. All right, a few more exercises. So here's the DNA. Go ahead and do the mRNA and the amino acid. The wild type, this means this is the normal DNA sequence. And then here are 3 different mutants. Okay, so the mRNA of the wild type is UGC. Mutant 1 is UGG, UGU, UGA. And then come back, and so for example, UGA was a stop codon. I don't remember what the other ones were. One of them was UGG. Anyways, here's what it shows what they are. Okay, so compare them with the wild type. Okay, so the wild type produces cysteine, mutant 1 produces tryptophan. So that's a missense mutation. It's an amino acid substitution. Depends where on the protein it is and the difference between the side chain of tryptophan versus cysteine to how harmful it will be. So it's kind of in between. You can't really predict how harmful it is unless you know where in the protein chain it is and what the protein does. Okay, number 2, you start with cysteine. Both UGC and UGU are cysteine. So it's a silent mutation. It's the identical amino acid. So it is a DNA mutation, but it's completely harmless. And number 3 is a nonsense mutation. So once you've told it to stop, if it's early in the protein, now instead of having a 146 amino acid protein, maybe you have a 2 amino acid protein, it's going to make a non-functional protein. So that's going to be the most harmful. Okay. Blank cannot leave the blank. So DNA cannot leave the nucleus. Okay, blank, copy the genetic information, moves to the cytoplasm, where the 2 units of the cytoprotein synthesis attach. So, copies the information. Messenger RNA moves to the cytoplasm where the 2 units of the ribosome attach. Okay, adapters in translation, that's got to be transfer RNA molecules, carry amino acids. Amino acids are monomers of proteins. And the finished product is a protein, a folded up polypeptide. Okay? Okay, and this you kind of go back and forth. For the DNA, A to T, C to G, C to G. Okay, for the DNA to the RNA, C goes to G. Here, A came from a T, a U came from an A. And the anticodon to the codon, U goes to A, U goes to A. So you can start going back and forth with the DNA and the RNA. Back and forth with the DNA and the RNA. And then just make sure you read the amino acid from the mRNA codon. We just don't read it from the tRNA codon. So AGA, and see what AGA is. AGA is arginine. So AGA is arginine. And this one, the UCG must be serine. And I think the AUC was isoleucine. I have to look.